Now scientists have discovered recently a different type of secondary structure in the case of collagen, and that is called, that's why, as the collagen helix. Now the reason is that collagen has a very large amount of glycine, proline and hydroxyproline. So because these are helix breaker, that's why these type of secondary structures are not possible in the case of collagen. Now please remember, collagen is made up of 3 polypeptide chains, and these chains develop left hand, that means anti-clockwise type of turning, and develop hydrogen bonds between the 3 chains, and this is called as the collagen helix. So there are 3 types of secondary structures possible. If it is a monomeric chain, alpha helix. If it is a oligomeric chain, either it will be beta pleated form or it can be collagen helix type of form. Collagen helix is different because of the abundant presence of the helix breaker. Beta pleated will have very few or zero helix breaker, and alpha helical structure is only possible in a single chain of polypeptide. So remember, the bonds which are formed in the secondary structures are hydrogen bonds. So this structure is developed, this structure has peptide bond formation, this structure has hydrogen bond formation. It can be in the same chain or it can be in 2 or 3 different chains. Now the next degree or the next level of the structure of protein is the tertiary structure. What exactly is tertiary structure? Now this whole thing, okay, one more thing, remember, every protein will have a primary structure. Every protein will have secondary structure. Now remember one thing, but tertiary structure is not found in every protein. That means that there are some proteins which are called as fibrous protein. They look like fiber, like for example, you all know about fibrin. Fibrin is required for clot formation. So all those fiber type of proteins which are fibrous in nature, and they are not like enzymes or hormones. So those fibrous proteins will have only how many structures? 2 structure, primary structure and secondary structure. They don't have any more other level of structure. But when it comes to globular proteins, so proteins are of 2 types in shape, fibrous and globular. If a protein is round or spherical in shape, it will have tertiary structure also. So please remember this much, that the fibrous protein will have only primary structure and secondary structure, but other proteins will even have tertiary structure. Now what is tertiary structure? The whole fibrous thing, because these are going to be a long thread type of structure, though they are coiled like this whole thing is going to turn around one more time. So now the folding and the bending of the secondary structure of the polypeptide is going to be there, so that a very compact 3-dimensional structure is formed, and this structure will even have the active site. Now what is active site? We all know a lock has a place where the key has to be inserted, and then only lock can be opened, and this the place where the key is inserted is very much matching with the shape of the key. In the same manner, if we talk about enzymes, enzymes have active sites, and in those areas a particular substrate or reactant has to attach and a reaction takes place. So active sites are synthesized by tertiary structure. That's why I told you in the last page, if you perform heating of a protein, reaction with a strong alkali, reaction with a strong acid, etc. So which structure will be damaged? Tertiary structure. And when you damage the tertiary structure, the active site is gone and enzyme becomes denaturation. It can be permanent denaturation or it can be a temporary one. So now the active sites are stabilized with the help of which bonds? So the answer I have mentioned here. There is possibility of development of hydrogen bonds. There is possibility of development of ionic bonds, even hydrophobic bonds, even there is possibility of van der Waals forces. We have learned all these things in the junior classes. Disulfide bonds. So there are many bonds which will develop, and they will cause the whole chain, which has become a secondary structure now, to undergo such a type of coiling that a wool-like round structure is formed, which will have its active site prepared. An active site is always different for every enzyme, just like every lock will have a different type of mechanism inside to open. So in the same manner, every enzyme will have a different type of active site, and that will ensure that the enzyme is reactive or not. So various levels you have understood now. Now if a protein is monomeric, it will have primary structure, it will have secondary structure, it will have tertiary structure. If it is a fibrous type of protein, it will only have primary structure and secondary structure. If it is a globular protein, it will have primary structure, secondary structure and tertiary structure. Remember, but if there is a protein which is oligomeric in nature, then there will be another degree of or another level of the structure that is called as the quaternary. Quaternary is the fourth level. So what is quaternary structure? It is found only in the oligomeric or you can say the multimeric type of protein. So when there are large number there are few number of protomeres. What are protomeres? All those chains of polypeptide which join to give rise to a quaternary structure. So if there is a protein which is oligomeric protein, it will have more than one chain. So now only those proteins which have more than one chain can have quaternary structure. So what happens in quaternary structure? All those single chains, like for example, see, every quaternary structure will also have these level of structure. So let's take an example of hemoglobin. Hemoglobin will have 4 chains. 2 chains are alpha, 2 chains are beta. So both alpha and beta chains will first of all have their own primary structure. Then they will start bending and all to form a secondary structure. Then they start folding to form a wool type of structure. Now how many wool type of structures will be there in case of hemoglobin? There will be 4. Now these wool-like structures are going to come close. How many? 4 of them. So 4 of these structures will come close and develop again bonds. Now these bonds will cause the development of the quaternary structure. So for example, hemoglobin has 4 chains, 2 alpha, 2 beta, and please remember all of them will have particular type of primary, secondary and tertiary structure. So for example, alpha chain has 141 amino acids and beta chain has 146 amino acids. So all these things are fixed, and this will ensure that the protein develops a quaternary structure.